it was the disease and the metabolomics. So we covered many series of the lectures in which you can understand that whatever the situation that is comparatively with respect to the control, we can use the metabolomics. There is a lot of the groups in India also, whoever is doing the research in the metabolomics with respect to the disease. So someone is the IIT Mandi, a few in the JNU, and a few in the DU also. So today I would like to go one step ahead of this metabolomics and the metabolomics and the disease. We would like to find out how can we use the metabolomics for the biosecurity actually. Diagnostic purpose, biosecurities and the responses. So this lecture is small lectures, but it will be very informative so that you can understand how it is useful for us. So we would like to, you know very well about the biosecurity. So biosecurity is associated with many phenomena, and these four phenomena are the most important for all of us. That is the environment, economy, health, and social and the culture value. So when this, we have to protect all these points actually, and you know GM crops are here, but somehow we try to create the awareness among the populations of our country, so without making any interferences with the social and the culture value, we would like to incorporate a few GM crops in our country. So last few years, you saw the government of India also release the guideline. So GM crops is also a concept that is associated with the biosecurity. Health, you know very well, if we are not healthy, so how our population is healthy. And then what are the metabolites which can give us a good profiling so that we can maintain the biosecurity in terms of the health of the individual of our country. Economically, of course, metabolomics is giving a overview in the biosecurity with respect to the economy. Environment, you know very well, sometimes some invasive species are present and sometimes there is another components are present which, if we know this metabolites is due to, that is present in a particular invasive species, so we can use this biosecurity for using the metabolomics for the protection of the environment also. So focus is that here of this presentation, focus is that how we prevent the exotic organism from invading or establishing. You know many plants are not native of our country and many invading or plants come from other countries in somehow in our country, and now we want to protect our area because their growth is where invasive species has very good growth and they can become the dominant after some time. So this is the strategy which we can use to protect our native plants from the invasive species. So importance is that we would like to provide the safeguards, indigenous species and valuable crops and the livestock. So this is the points, and rising challenge in the biosecurity. So they are the few challenges we have to face when we have to go ahead with the biosecurity. So that first one is the increasing the results from the insect pest as well as the pathogen basically. So what is the meaning of it? Actually, when we are using the pest, whatever the pesticide we are using, so pathogen are going to kill. And that's why we are decreasing the population of the native organisms many. Growing pressure on the biosecurity system, this is another challenge. Policy implementation is, of course, it is the major challenge that is associated with the policy makers to implement the policy at the ground layer to make it successful. The eradication and the management. So now I'm coming here on that, how can we explore the metabolomics for the plant health biosecurity basically. So beyond species identification. If in one class during the first lecture of mine, you know that I told that metabolomics is useful for the species identification. So if you think that it is only up to the species identification, so might be we have to think beyond the species identification. We have to find out the biosecurity assessment of the different plants with respect to the insect pest as well as the plant pathogen. So many plant pathogens, they come on a particular plants for a particular activity. You know that this citrus plant has many components and they are egg-laying behavior of many butterflies are only due to the presence of some specific pollenions which are present in the citrus plants. So because they lay their eggs there, and finally the newborn small insect, small bean of the insect can protect itself by inject those flavanol, and they can protect them from the different type of the another pathogen and the other birds also. So what is the meaning of that? If you know that these are the metabolites which are responsible for the pink bollworm that is labeled as a sundi in this area, if you know this is the metabolites, that's why those insects are coming on the cotton flower. So we can use it for the biosecurity purpose. We can protect our crops. In the another way, if we can also protect other organisms which are generally killed or died due to some specific pesticide. So what pesticide is working on which insect, and so we have to make pesticide friendly with some another insect which are not harmful for the plants also, and it can give the informed critical biosecurity responses. So we have to think the role of the metabolomics beyond their species identification. The another one is point here, what are the latest application of the metabolomics. So here you can know that this is a comparatively fingerprinting for the diagnostic purpose. You know some disease plant and some healthy plant, so how we can go ahead with the comparatively analysis. So comparatively analysis always gives us the overview of many biomarkers, and these biomarkers, upregulation and downregulation of these biomarkers are enough to show the early pathogenesis state, early infection. So you can prepare the plants and the crop before you get the hug loss. The metabolic profiling to uncover new association between the metabolites and the phenotypes also. If you identify some metabolites which is responsible for a specific phenotype in a particular plant during the disease condition, so you can go ahead and you can use the same thing for the association, and by using this association, up regulation and the down regulation of metabolites is directly associated with the phenotypic appearance of those plants. This case is similar to any disease when come in our individual level. So if one person has some infection, another person is healthy, so you can find out both has the different type of the phenotypic appearance. So that's why the same thing is happening in the plants, and it is useful not only for the plants, it is also useful for other organisms. Insight from the current research is there that this metabolomics is applicable in the diagnostic as well as the fundamental investigations, and its potential both in the insect and the plant pathogen detections also. So you know very well now artificial intelligence is come up in the picture. So if you identify some spectrophotometer which can detect that specific metabolites, so you can scan and you can say that this plant has this infection without touching that plant. So this type of the things you can do in the whole field or on the whole area also, if that metabolites is giving some spectra which we capture by some spectrophotometers, and then it is easy without making any loss to the agriculture, or in the near future these crops will get some infection. So we can get it before we get hug loss. The another points is that focus area. So these are the two focus area of the metabolomics we would like to explain here. That is the insect biosecurity threats, and second is the plant pathogen detection. So I don't know what is happening with this speaker actually. Sometimes it become okay and sometimes it switch off. So I'm coming here. That is a new area. That is the entometabolomics. So entometabolomics apply to the insect biosecurity. And yes, entometabolomics involve the metabolomic analysis of the insect to study traits like species. I don't know why. This is the power. Provide critical insight into the invasive species characteristics such as their geographical origin and host plant at the risk, enabling effective biosecurity. So basically, this is another new area where we have to think about it. That is the entometabolomics. नीलेश यहाँ देखना बैटरी पड़ी है नहीं? वो पड़ी हुई है ना? Hello? Maybe there. This is the battery issue. Now it is working, but give me. Maybe there is some issue with the battery. दो biochemical information about the invasive species such as their geographical origin, life stage and host plant which are at risk and due to these biosecurity measures. So we have to do this biosecurity measure and we can get such type of the information to protect our native plants from the invasive species. So application, first one is that I told you about the species identification. It distinguishes cryptic species and correlating metabolite profiling with the genetic differences. Example include the separation of the cryptic fly and the termite species based upon the CHC profiling. So try to understand, we can separate some species also on the basis of a particular chemical profiling. So now I am coming on the another parts, that is the sex and the age differentiation of the insect basically. So it's very difficult to differentiate many insects based upon their gender as based upon their age. So this metabolic profiling is giving the crucial information to understand the population dynamics, metabolomics, can differentiate the different genders even in the dimorphic species also. And this is also changed with the based upon the age and the sexual maturity. You know, the metabolic profiling of the newborn baby and the adult one and the twins, this is totally different among us also, human being. So you can understand in the plants this thing is totally different, and even the insect also this thing is totally, you can differentiate only on the basis of the metabolic profiling. Population structure and the reproductive potential also. Post-mortem analysis, so many times what happening actually, you know, many plants, if you put a plant today and if you are taking the analysis after 5 days, so you don't know how much it is the fresh. So after 5 days, most of the metabolites, whatever the substrate is present, they were utilized by some biosynthetic pathway, and now you can identify this plant is 5 days old picked. The same thing happening in the post-mortem of the individual. So you know very well what happening actually in post-mortem. We can easily identify what is the actual time and the death of any individual on the basis of some specific change in the parents, some specific change in the organ structures, and on the depletions of the different metabolites. So similar things can be carried out in the insect also. Here we can do the post-mortem based upon some metabolic profiling which are going to decrease or upregulate with respect to the actual death timing. And this can be past timelines also. Geographical origins, yes, some insect, some organism and some individual. We collected here from the different geographical area of our country. We have the different behavior also. Aggressiveness, softness, humbleness, and way of talking, skin colors. These are the totally different from each other. So if you see some insect is coming from the Pakistan, okay, by the fly, by the air current, and some insect which are already present in the Madhya Pradesh. So they have the different type of the subspecies, and we can differentiate this one is coming from the Madhya Pradesh and this one is coming from the Pakistan. So on the basis of the metabolic profiling of those things, on the basis of that we can track their movements also, how they come here and what is the real issue, that what the things which enforce them to move from the Pakistan to the India and from Madhya Pradesh to the Punjab also. The dietary and the post-plant analysis is another area which is also considered into the entametabolomics, that differences in the insect diets give distinct metabolomic signature, adding in determine host plant and the feeding pattern. So this plant pathogen detection can be done by what is the plant defense actually. Plant protect itself from the different infections by using 3 different strategy. The first one is the cell wall. Cell wall is the primary defense of the any plants, and production of the secondary metabolites is the another one, that is the phenolic components, and generation of the reacting oxygen species. So this is the third part. So these 3 mechanisms are basically protect the plant from the different type of the infections. Now challenge in the detections. So what are the things which we have to prepare ourselves during the detection of those infections? So first one is the pathogen suppressing plant defense basically. Pathogen only when enter into a plant, it try to establish itself in the plant when the defense mechanism of the plant is compromised. And sometimes they overlap with the stress-induced metabolic changes also. So application of the metabolomics in the biosecurity is the pathogen detection is one of them. temporal dynamics is the another one, and sampling strategy that we have to understand to take the sample from which part. I know we are working on the cotton plants, and there was a problem to detect the infection of a virus in the cotton plant. So we collected the sample from the different part of Haryana, Rajasthan and Punjab. So it was very difficult to get plant phenotypic appearance to prove that they are the infected samples. But when we collect from the leaf, so we are not able to detect them by the normal PCR. So what we did, we remove the veins and use the juice of the vein to identify that there is a lot of the infection of that particular virus in that leaf tissue. So what was the reason that phloem is something that is moving from one part to the another part, and this is a site where this virus is multiplied too much high. So because it has so many nutrients. When cell is bred, so they releases so many viruses, and these viruses move by using the phloem from one side of the infection to the rest of the plant. So this way we focused ourselves only on the phloem sap to identify the infections in the cotton plant. So the same thing is here. What sample strategy you have to use? Sometimes you have to use with the phloem, sometimes you have to use the leaf tissue also. So this is a case we are taking here. This is the strawberry has the infection, and this is the botrys cinerea showing the distinct grey mold. Both this fungi is the same, but it has, you can find out they are giving the different type of the symptoms here. The first one is in the strawberry, and second one is going the grey molds actually. So on the basis of that, it means they have a different type of the defense mechanism, and that's why we have to use the metabolomics for this type of the work. Here I am explaining the tools and the techniques which we have to think to take for this entometabolomics, that is the mass spectrometry in the metabolomics, high throughput phenotyping, because you have to correlate both of them, otherwise it will be difficult for you. How you go ahead? The first you have to use the latest metabolomics techniques, and secondly you have to do the high good phenotyping of that area. So automatic systems you have to use sensor imaging, so that you can make a tool on the basis of the metabolic profiling by using some tool. So challenge are the baseline variations, diurnal cycle, stress, soil and the seasons, because every season has the different type of the infections. Soils also impart the type of degree of the infection, stresses, then diurnal cycle, day and the night. Differential native and the exotic pathogens, you can also differentiate what is the indigenous pathogen and what is coming from the outside. So future direction of this type of the work is that integrating metabolomics with the genomics and the proteomics, and developing robust field applicable diagnostic marker. So now these are the pathogen detection cases with respect to the metabolomics. You can find out the strawberry here, citrus plant, winter wheat, fungal pathogen and the viruses. So they use the Cholesterium theobromicum, and they are the strawberry leaves, and the metabolic profiling is they detected 4 days before the symptoms. So if you do the metabolic profiling before the 4 days, you can easily identify that this is the this particular plants is going to get the infections, and this can be used by the GCMS, and temporal profiling crucial for the early detection. Second is about the citrus plant. So Candidatus liberibacter species is the pathogen. So citrus, that is only limited to the phloem, and it is a specific metabolomics profiling in the phloem tissue only. LCMS is used here. This is the tissue specific sampling critical for the accurate detections. The winter wheat is basically, pathogen name is the smut fungus. It is a winter wheat grain, and lipid signaling molecules, that is the prostaglandins, upregulated during the infections. We use the comprehensive overview to identify which lipids can be used, the potential diagnostic marker for this particular activity. Fungal pathogen is the Fusarium culmorum, and that is the general host pathogen system. Insight into the host pathogen interaction based upon the LCMS with identification of the unique metabolic profiling. This is potential integration of the metabolomics with the proteomics and the genomics. Viruses basically viral coat protein, peptide fragment of the viral coat protein identified using the mass spectroscopy, and MS can identify what type of the infection is present in that particular case. So metabolomics by the NMR in the plant disease is also used. So this is an example of this particular citrus plant, and they are fruit infected by the Hoang Long Bing disease. So it is a severe citrus disease causing significant economic loss globally and transmitted by the psyllids infectant that Corina citri on through the infected graft. So challenge, there is no cure available till today. The control strategy is the removal of the infected plants and vector controlling using the insecticides. So these are the two part of the controlling strategy. Also issue with the insecticide resistance and the environmental concern. So that's why you understand NMR was used for this purpose actually. So visual inspection for the late stage symptom is a major issue, and confirmation via PCR and the qPCR. Limitation, long incubation periods, 6 months, the same in the cotton, it's 90 days, high post and the time invasive nature of the PCR, inability for the large scale early detection. So role of the NMR in the metabolomics for the HLB diagnosis is the advantage of the NMR in the plant research. It was used basically, suppose you are using the NMR for this particular work. So rapid minimum sample preparation, high reproducibility, useful for identifying the biomarker under varying conditions. Methodology, we have to use 1D, 2D, NMR used for the identifying metabolites. Chemometrics tool like the PC and the PLSD applied for the biomarker analysis. So K finding in the biomarker identification by doing this method. So healthy insect affected and the sick leaves separated using the PC and the PLSDA. K spectrum region for the biomarker is identified with respect to these area. So you can identify that these are the area we have this delta 9 to 8.4, 8 to 6 and 3 to 2.8. So you see that these NMR spectra, and here you can identify these three spectra which are just like the biomarkers. And these PCA plots, PCA and the PLSDA plots is reflecting the dissimilarity between the infected and the non-infected plants. So advantage of the biomarker based diagnosis is that it can be early and the precise disease detections, reduce the reliability on the expensive and the time consuming PCR, identification of the plant stress response in the metabolomic changes we can understand, potential for the developing diagnostic kit to the glucose kits, just like a glucose kit. So comparison with the root metabolomics investigations, so root showed the advantage feature of this particular disease after a long-term infections, and leaves provide basically clear insight due to the immediate metabolic interaction. This infection comes in the root very late, but it comes in the leaves very soon. So particularly of the leaf sample for the field diagnostic is used for this purpose. So potential biosecurity applications, here I am explaining that is the detection of the GMOs. So you can identify the GMOs also by using this NMR metabolomics. The another is the herbicide resistance monitoring. So you can detect resistance mechanism affecting weeds in the crop management. Require large sample analysis, and the advantage is the chemometric tools. Phytosanitization and the quarantine is the potential for verifying their devitalization and the fumigation treatments. Limited current application in the phytosanitation assurance. So these are the potential application of the biosecurity application, and we already proposed the example by which you can use the NMR for the disease plants and the non-disease plant to identify the biomarkers in that particular case. So here I am just going to finish, and then Ritu, please. Now if you are, you can. So now conclusion is that there is emerging biosecurity challenges you have need to take care. Potential of the metabolomics is always giving. You can get the comprehensive information from a single analysis only. Species, sex, time, death, host plant, parasitism, and origin of the insect can be easily identified using the metabolomics. Limited but promising application for the plant pathogen. Future research needs the significant efforts required for the practical implementation. Focus on the extending current research to the biosecurity species applications. So long-term benefits is the complement existing taxonomy and the molecular diagnostics, and enhance scientific understanding of the biosecurity threats and the plant interactions. So yes, it was a very short, but it gives the comprehensive overview where we can use this entometabolomics, where we use the metabolomics to protect the native plants from the invasive plants, and if there is some disease and it is coming late, so we can use the metabolomics to early detect the before it come in the form of a symptom, you can detect that particular infection only just to the unaltered metabolites also. Yes, I would like to appreciate many thanks to all of you for this listening of these lectures with very patience. I would like to acknowledge the Gyan Administration and the Ministry of Education, India, for to fund sponsor this program. And yes, metabolomics, these are the two papers we considered for this particular proposed work, that is the metabolomics by the NMR spectroscopy in the plant disease diagnostic, and second is the metabolomics for the plant health, biosecurity diagnostic, and the response in the sustainability. So now Ritu is coming for a half an hour and she will explain what type of the work she is doing and how you can use the interpreted data basically. So Ritu, please come. She is working on the metabolic profiling of the different seed development of the CP in Desi and the Kavli. So yes, it is something that is associated with the metabolomics. We are using 4 to 5 different techniques. First is the GCMS, second is the LCMS, then NMR, and then FTIR. So and HPTLC also. We are using 5 different techniques to identify few unaltered metabolites. Yeah, please. The professor जी, come this. आप मैडम को फोन करें.